So this particular video is going to just be this one slide, and it's a bit of a review. That being said, I don't think you should skip over this one, even though I'm reviewing stuff that I just covered, because this is going to put everything together in context. And once I'm done putting everything together in context, I'll talk about like what about this stuff I really do want you to know for things like exams. So what I want to do is I want to talk about transcription translation as it occurs in the cells themselves, so you understand how all of this stuff that I just covered on the last two videos actually works. And so what I want to do is I'm going to start off by giving you a DNA sequence. And here's the DNA sequence I'm going to give you. The DNA sequence is going to read TAA, TGA, CTG, CCC. Alright, so the first thing that I wanted to say about this DNA sequence is that, of course, remember, DNA is double-stranded, and I'm not going to deal with that in this particular video, but what you can do on your own free time, or pause the video right now if you want, what's the sequence of the other strand of DNA? Go ahead and figure that out. But what I want to do is to figure out how transcription works here. Let's do transcription and convert this DNA molecule into mRNA, where we create an mRNA from this molecule of DNA. So let's go ahead and do transcription. So going to use base pairing rules. So T pairs with A pairs with U, A pairs with U, A, C, U, G, A, C, G, G, G. And remember, in mRNA, 3 bases in a row is called a codon. And remember, this particular mRNA has 4 codons in it. So I've just underlined each and every codon. So a single mRNA molecule can have multiple codons, like what you see here. Basically 1 codon per each amino acid that's going to be found in the protein. Next up, the tRNAs. Now remember, for each individual codon in mRNA, a different tRNA will bind to it. So let's do this one at a time, 1 codon at a time. The first codon here reads AUU. So what's going to be the anticodon for the tRNA? Well, the anticodon is going to be UAA, because that forms a base pair with AUU. And of course, there still is the rest of the tRNA. Let's go to the next tRNA. So the next codon reads ACU. What's the anticodon for the tRNA that binds to that codon? Well, it'll be UGA, because UGA base pairs with AGU. And of course, we have the rest of the tRNA. The next codon in mRNA reads GAC, so the anticodon in tRNA will read CUG, and that's the rest of the tRNA. And lastly, we have the codon GGG. The anticodon is going to be CCC, and of course, there's the rest of the tRNA. These lines here that I've drawn when I've been saying the rest of the tRNA, that's just the rest of the tRNA sequence. I really don't care what those nucleotides are. I'm just drawing lines to show that this tRNA is, of course, much longer than just these 3 base anticodons. Yes, and remember, those 3 bases in a row are called anticodons. So this mRNA had 4 codons, so there are 4 different tRNAs, each with their own anticodon, that bind to this mRNA. By the way, one last thing, DNA to mRNA, of course, that's occurring at the nucleus. That is called transcription. mRNA having tRNA bound to it, that's a part of the process of translation, and this is occurring in the ribosomes. So don't forget that. Lastly, the other part of these tRNAs that I didn't mention is that at the top of them is an amino acid. So what are going to be the amino acids for these tRNAs? Well, let's think about it. Let's try to figure out what the first amino acid is going to be. I'll give you a second, or pause the video to get out your genetic code and figure it out. Okay, so hopefully you said ILE or isoleucine. So how do I know it's ILE? Well, remember, the genetic code uses the codon information. So if you look up AUU on the genetic code, you will see that AUU codes for the amino acid isoleucine. The next amino acid is going to be threonine, or THR. And the way to figure that out is by looking at the genetic code. ACU, the codon ACU, codes for threonine. GAC codes for the amino acid asparagine, or ASP. And finally, the codon GGG codes for the amino acid glycine, GLY. So what you can see happening here is that when these tRNA molecules bind to the mRNA in the ribosomes, the amino acids in this protein are now all lined up, kind of like ducks in a row. And so the last thing that happens here to make the protein is that these amino acids chemically bond to each other and break off from the tRNA. Now there's still the rest of the tRNA molecule, but the amino acids are no longer attached at this point. And what we have now is a fully formed protein ready to go. So this is the basic concept of how this process works, of how transcription and how translation works, to start off with a molecule of DNA, to start off with a gene, and end up with a protein. So please make sure you review this video, because I think this video and looking at this final image here kind of shows you biologically what happens each step of the way, even though on paper, that genetic code is a really nice little bit of a shortcut here. So based on this, what do I expect you to actually be able to do on a homework question, on a lab worksheet question, on a test question, for example? Let me cover this. Number one strand of DNA to the other strand. You should know how to do that. That's just base pairing rules. Next up, DNA to mRNA. You should know how to do that. That's just base pairing rules. Codon to anticodon. You should know how to do that, because that is also just base pairing rules. And also codon to amino acid, because that's just using the genetic code, which you should know how to do based on the previous video that we just did and this video here. So what am I not going to ask you? Well, I'm not going to ask you to go from the anticodon. So for example, I'm not going to say, oh, go from anticodon directly to amino acid. I'm not going to do that, because the only way you can really do that is to go from anticodon to codon, and then from codon to amino acid. And to me, that just seems like jumping through extra hoops for no real good value or reason. So I'm never going to ask you to do that. So this is what I'm going to be asking you to do. Go from one strand of DNA to the other, go from one strand of DNA to mRNA, go from codon to anticodon, go from codon to amino acid. Now, going from codon to amino acid, of course, you are going to be using the genetic code. I'll let you know right now that on any test question that asks you to use the genetic code, I will give you the genetic code in the test question itself. You will not need to try to memorize the genetic code. I mean, there's no point in that. I don't even have it memorized. I have to look it up every time I use it. So I don't expect you to memorize it either. But you do need to know how to use the genetic code correctly. And so how do students mess up using the genetic code? Well, the most common error that I see is that students think, oh, to use the genetic code, I need to use anticodons to get to amino acids, and that's completely incorrect. Remember, the genetic code is dealing with codons, not anticodons. So if you have the mRNA sequence with you already, and you have the genetic code, you just look up that sequence 3 bases at a time, and it'll spit out the amino acid sequence of the protein. So that is probably the most common error that I see when students are using the genetic code, that they use the wrong sequence. Remember, the genetic code is always showing mRNA sequence. So going from mRNA to amino acid is always using the genetic code. Whoops. So make sure that when you're using the genetic code, you're using the mRNA sequence to go directly to the amino acid sequence of the protein. That to me seems to be the major error that a lot of students make. So just make sure that you avoid doing that. Besides that, I think that's really about everything that I wanted to say here. You know how to go from DNA sequence to DNA, from DNA to mRNA, from codon to anticodon, and with the use of the genetic code, you can go from codon to amino acid. And I know I'm repeating myself here because I'm just really trying to hammer down and make sure that everything is really clear here with everything that you need to know about how to do transcription and translation, for any test questions that I throw out at you. All right, so that takes care of this. And in the next lecture, in the next video, we'll talk about how this all gets messed up.